DNA, you've got lots of it in your cells, but sometimes in the lab you've only got a small sample to work with. So today we're learning all about amplifying whatever DNA we have using this process called PCR, or polymerase chain reaction. There are lots of reasons why you might only have a small amount of DNA to work with. So for example, when we're looking at like evolution, you might have found a fossil that has a very small amount of viable DNA in it. So we want to amplify what we have so that we have enough for analysis and looking at evolutionary relationships. Or you could be a forensic scientist and someone has committed a crime. They've left a small amount of blood at the crime scene and you need to work out who left the blood. So you're going to need to copy that DNA and have lots of it on hand so that you have enough for analysis. Now there are lots of different ways you can analyze that blood, but that's like future video topics there. With DNA amplification, we are taking a small sample of DNA and making lots and lots and lots of copies of it. We're able to amplify DNA because of the unique properties of DNA itself and its structure. DNA is a double helix. It's made up of two strands that are complementary to each other. So once you have the code on one strand, you have the code of the other strand as well, because it's just going to be the opposite. So when we have a small sample of DNA, essentially we have two templates that we can use to build more and more strands of DNA. Polymerase chain reaction was invented by an American scientist named Kerry Mullis, and he discovered this in 1986 as a technique, and he did win a Nobel Prize for this in 1993. Polymerase chain reaction essentially takes a single isolated bit of DNA, so there are lots of different ways we can isolate DNA, but once we have just pure DNA, we are going to chuck it into a test tube with a few other ingredients. We are going to put free nucleotide bases, and these are the little bases that build up a long strand of DNA. As well as nucleotide bases, we are also going to be adding these things called primers, and we'll get to that later on, and this really important enzyme called DNA polymerase. Now, it's an enzyme that builds DNA. We have DNA polymerase in our own cells, and that's what copies our DNA each and every time we're going to undergo a process of mitosis. You can tell what DNA polymerase is going to be just kind of by its name. So A's at the end, you can tell what we're looking at an enzyme. It's a really common suffix that we have at the end of different enzyme names. And we're looking at DNA polymers. So building a big molecule made up of small building blocks, which are monomers. So joining monomers together, in this case nucleotides, is going to give us our big macromolecule, which is DNA. Each and every cell out there is going to have its version of DNA polymerase. So obviously human cells will have our own human DNA polymerase, but if we use this in this particular process, things aren't going to work. The DNA polymerase that we use in PCR is not human DNA polymerase. Our human DNA polymerase would essentially just denature at the high temperatures that we actually undergo the process of PCR. So what we are using for actual PCR is what we call Taq polymerase. Taq polymerase comes from Thermus aquaticus bacterium. So Thermus aquaticus, put that together, you get T from Thermus aquaticus, you get Taq. It is a DNA polymerase that we isolate from bacteria that lives in super hot geothermal springs. Taq polymerase, as a variant of DNA polymerase, can survive super high temperatures, and their ideal working temperature to build a strand is much higher than the 37 degrees Celsius that human DNA polymerase is going to work at. DNA polymerases all work in one particular direction, so they're going to work in the 5' to 3' direction. So they're going to take our template DNA strand, read it, and build 5 to 3. In order for PCR to work, we are going to need to basically chuck everything that we need into these little test tubes. So we're going to put our DNA template, we're going to put primers, we're going to put free nucleotides and our Taq polymerase, as well as some buffer solutions. We're going to leave these in a machine called a thermocycler. A thermocycler is going to basically change your temperature at different points, and it will cycle and go through this process again and again and again. We have 3 stages of PCR and 3 distinct temperatures where we're going to have the PCR reactions occurring. The first step of PCR is denaturation, and this happens at 95 degrees Celsius. So we heat up everything in our little test tubes, and when that happens, our double-stranded DNA that we want to copy is heated up so much that we actually break the bonds in between our nucleotide bases. So we break the bonds between A and T, C and G, and what we get are 2 single strands of DNA. 95 degrees Celsius is also super high in temperature. So this is why we need Taq polymerase. We need a heat-stable enzyme to build our DNA, because if you were to use human DNA polymerase at 95 degrees Celsius, it's definitely denatured and it's essentially going to be useless. We have an optimal temperature of 37 degrees Celsius, get a bit too high out of that range and the protein's no longer functional. So Thermus aquaticus provides a really nice heat-stable protein that's able to survive this 95 degrees Celsius denaturation stage. Denaturation also doesn't take that long. We're heating everything up to a really high temperature for a short period of time. So depending on the length of your DNA you have, it might take seconds. In general, we're looking at about like 30 seconds for most strands of DNA though. Once we've got our 2 single strands of DNA, we're going into our next stage of PCR, which is annealing. Annealing happens at a temperature around 50 to 55 degrees Celsius. So it's cool enough that we can add these things called primers, but it's not so cold that our 2 strands of single DNA are going to form together a double strand once more. In annealing, we're going to be using these things called DNA primers. A primer is a short section of, in this case, DNA that's going to be complementary to usually a region of DNA near the start of a strand. So if we know like common start sequences, we're going to have a short bit of DNA that's complementary to it. It'll bind on, and we know that this is something that our DNA polymerase later can recognize and use as a starting point to actually build our strand. DNA primers can vary in size, and you can actually make really specific DNA primers depending on like which section of DNA you want to copy. So let's say we actually want to find a particular gene of interest. We know a little sequence that starts this gene. We can make a primer that sticks to that gene, and we know that we're always going to amplify that particular section of DNA. We're always going to have the gene of interest copied. DNA primers require a relatively lower temperature in order to bind themselves to our template strand of DNA. If we'd kept everything at 95 degrees Celsius, that's a high enough temperature to break apart our double helix. It's a high enough temperature to break a lot of bonds between adenine, thymine, guanine, cytosine. So our DNA primer has like no chance of binding itself to the template strand at 95 degrees Celsius. But at 50 to 55 degrees Celsius, they're able to come in and bind to our template strand quite well. In the annealing stage, you actually need 2 separate primers that are added to our mix. So this is all added at the beginning. You're not like stopping things, putting stuff in, and then like going in between each cycle. All this stuff is ready to go in our test tubes. We need 2 separate primers because when we separate our DNA, they're mirrors of each other. The template on the top strand is going to be complementary to our template on the bottom strand, which means when we're building something, we're going to need a different start sequence for each strand. DNA polymerase only works 5' to 3', so when we do separate our double strand, we've got a 5' to 3' sequence that looks different on one strand compared to the other. So we have 2 primers. We have a forward primer and a reverse primer, and they're going to bind to their respective complementary strands. Once the primer is in place, we then can heat up everything so that our DNA polymerase can come in, bind, and then do their thing of actually extending the strand and building our new DNA. Think of the primer as like that little metal bit at the end of like a zip that will go into the actual zipper? That's what a primer is. It's a little bit that is going to find our template strand and help feed it through our DNA polymerase later on. Annealing doesn't take that long as well. It takes like 45 seconds generally. Once we're done with annealing, we're heading to our 3rd and final stage of 1 cycle of PCR, and that is extension. For extension to happen, we need to raise the temperature to around 72 degrees Celsius, and at this temperature, our DNA polymerase is going to go around, find where the primers have bound, and then use that as a starting point to build our strand of DNA. It is going to read each and every base that it comes across and join a complementary nucleotide all the way together to form a long strand of complementary DNA. Once the DNA polymerase has reached its end, so the Taq polymerase has finished reading everything, it's gotten to the end, it'll fall off, it'll work on the next strand of DNA. Extension only takes usually like 45 seconds, and it will keep going as long as we have enough template strands of DNA, and enough free nucleotides. So once we get through our 3 stages of denaturation, annealing, and extension, that's 1 cycle of PCR. The benefits of a thermocycler is once we're done with 1 cycle, we can repeat it again and again and again and again. You can do this as long as you've got enough of the starting materials there. If you consider how fast the PCR process is, it's amazing how quickly we can have exponential amplification of our DNA. So after 1 PCR cycle, we're going to go from 1 strand of DNA potentially into 2 strands. And then after that, 1 more cycle, 2 strands becomes 4 strands, and it keeps going and keeps going and keeps going. You leave the thermocycler to run for an hour with like lots and lots of cycles, and at the end of that, you're going to have millions and millions of copies of DNA to work with. The benefits of PCR is that it's really quick and pretty cheap and accessible as well. So as long as you've got your starting materials, as long as you've got a thermocycler, you can actually get through these steps in a very short amount of time, have lots and lots of DNA to work with, and then you can do whatever testing you want once you're done with it. A modified version of PCR is actually also being used in COVID testing right now because it is so quick and cheap to run. So we have in COVID a retrovirus, we have an RNA-based virus. So what we're going to need to do is a little additional step where we convert that RNA into DNA. So to do this, we have a reverse transcription polymerase chain reaction, which is PCR, but to start, we need to convert RNA into DNA. So if you do have COVID, you're going to have the viral RNA that we're targeting. We know what we're kind of looking for, so we're going to put a little primer that binds to that RNA, and we're going to be using this other enzyme called reverse transcriptase, which we see in a lot of viruses that are RNA based. Reverse transcriptase's job is to actually take RNA and convert it into a single strand of DNA. So unless you've got the virus, you're not going to have the RNA, which we can then convert into DNA, and then that is not going to get amplified later on. If you do have the virus, then that RNA will get converted into a single strand of DNA, and when we chuck it into the PCR, we're going to actually see results because something is getting amplified. There are so many applications of PCR. Anywhere where you need to take a small bit of DNA and make lots of copies, PCR is the thing that we're using. Okay, without PCR, we wouldn't have been able to work out things like our evolutionary relationships with early hominins because, you might have found a little bone sample, but that's not enough DNA to analyze. PCR gives us enough DNA to analyze this stuff really, really accurately as well as we can anyway. PCR and its ability to amplify DNA has also been a massive game changer in forensics. So if you think about it, 60 years ago, you could have committed a crime, sprayed your blood all over the crime scene, and the cops would just be like, oh, that's gross. But they couldn't do anything with it. We didn't have enough knowledge of DNA or the techniques to amplify DNA or do anything with it in order to catch you. So now, you leave a single drop of blood, police are going to find you, they're going to catch you because we can amplify that blood and use your DNA to trace it back to you. It's amazing. This stuff is revolutionary, and we are learning so much because of our ability to amplify DNA. So I hope you learned a little bit about PCR, the 3 different steps that we have, and you understand a little bit more of how we can apply this knowledge in different technologies. We'll go into the actual nitty-gritty of the different technologies in future videos. So happy studying, and I'll see you next time. Bye.